Hello friends, how are you doing today? Again, this is morning. I'm just going for my duty. So meanwhile, I just want to talk to you. So we live in Hyderabad actually. This is the place where we live. So actually I'm waiting for my bus here, my medical college bus. Yes, my bus is here almost. Okay, so this is my bus. Okay, thank you. Okay, this is my lab, practical lab. My attender, hello Cheppu. So this is our practical hall for students. You can see the microscopes. By the way, I am a medical microbiologist. Okay. So students come for the practicals here. Here you see these are the actual reagents for some staining techniques that we perform in the laboratory, but I want to show you one thing here. You see this one. So this one. So decolorizing solution. Actually this is acetone. We are using this one for the nail polish remover. The best and the original nail polish remover. Actually this is a decolorizing agent. So generally I use this one for my nails. Okay, acetone. And this one is research lab where we can do some research work. These are all our instruments. So just we perform some research work here. You see the microscope and some of the instruments. And this one is a demonstration room. You see this one, students, they come batch wise, A, B, C, like that. One batch they go to practicals, one batch they come for demonstration, like that. Okay, we divide them. And you see here, board. All right. And this is museum. You see so many stuff here related to microbiology. Not only microbiology, this is of course related to medicine. Okay, the diseases, their treatment, how they cause infection, what is the pathogenesis, clinical features, stuff like that. So for the student purpose, we keep all of them like this. Here are some of the specimens you see. Okay. Actually we have to fill all the racks with the specimens. For now only we have few. And already we ordered some specimens. Big companies are there, they provide all these stuff. I want to show here the models of different organisms. Okay, their structures. Here you see this one is actually bacteria structure, and this one is also bacteria, and some of the bacteria they have extra structures like flagella. Okay, we call this one is flagella. These are the pili, stuff like that. I think if some medical students are there, if some science students are there, they easily understand what I am talking. Here the shapes of different bacteria actually. Rod shape, spherical and spiral shape basically. So you know the bacteria causing many infections in human beings. Here some viral structures. This is an embryonated egg where we can grow the viruses, because the viruses, they need some tissues or some living thing for their growth. So here this is the model, okay, how they grow in the embryonated egg. And this one is the coronavirus. You know coronavirus, everyone knew this, which is causing COVID. Internal structure of the coronavirus actually. This is the basic coronavirus. And this one is HIV virus. You see the bacteria is different, virus is different, fungus is different, parasites are different. There are so many microorganisms. They cause infections in human beings as well as in animals. Different viruses, they are in different shape, different structure. Their morphology is completely different. This one is rotavirus. Rotavirus is the causative agent for diarrhea. And poliovirus. Poliovirus structure is like this. They cause polio. And this one is helical virus. Actually, there are so many helical viruses. Again, there are some charts. Some scientists, their contribution, they did so much of work to establish all these things. The scientist plays a major role for the development of medicine. And here also you see some more viruses. So this one is the bacteriophage virus. You know, bacteria are small. Even the viruses are smaller than bacteria. This virus especially, they live on bacteria. They need bacteria for their growth. Okay, so they infect bacteria annamata basically. Okay, and this one is herpes simplex. So many. Adenovirus, hepatitis B virus, which is causing hepatitis, that is jaundice. Manamu jaundice ani pilustam kada, adannamata. Idi jaundice virus valla vastundi. Ikkada మనం ఇదేంటంటే ద లైఫ్ సైకిల్ ఆఫ్ పారాసైట్. అంటే పారాసైట్స్ ఉంటాయి కదా కొన్ని, మనం వామ్స్ అంటారు. ఎక్కువగా చిన్నపిల్లలకి అవి ఇన్ఫెక్షన్స్ వస్తాయి కదా. పారాసైటిక్ ఇన్ఫెక్షన్స్ ఆర్ మోస్ట్ కామన్ ఇన్ చిల్డ్రన్. ఓకే, హౌ దే గెట్ ఇన్ఫెక్షన్, దట్స్ వాట్ దిస్ model is showing. This one is one more parasite. Whenever they go into the water which is contaminated with the particular parasite, so this parasite actually pierces here and enters into the body, and then finally they cause infection like that. Okay, so this is our museum. And this one is library for students, and this is small library only for microbiology section, but there is a original library in other building, so that is very very big one. And this is gym room for students again. Okay, actually only few things are here, only for the students. Okay, the most important one is RT-PCR laboratory. This is comes under microbiology section only. Okay, where we are performing RT-PCR for COVID, HIV, hepatitis and everything. We are doing tests here, but nowadays we are performing COVID. Okay, this is the entrance. Inside you see the first one is shoe rack. Then here donning room. Donning room means we have to wear PPE, personal protective equipment here. Okay, so these are the PPEs, personal protective equipment. Before performing the test, the technician has to wear this. Another room, and this is sample receiving room where we can get the samples from here. Okay, somebody will bring samples, COVID samples or HIV, whatever the samples, they can just keep inside, and then a technician has to sit here, one technician has to be here, and they will just collect the samples from here. This is the door, they just open and they can collect. Because whoever brings the samples, they should not enter into the RT-PCR room directly. So everything is protected. And after collecting the sample from here, and there is another door here, the sample has to be kept inside. Now we are going to the other side, another room. Okay. Here. Okay. Here I told you the samples, whenever they collect the sample there, they have to keep here. One more technician has to be here, and they will collect the sample, and then they have to process here. This is the actually sample extractor. Whatever we need from the sample or the organism, we can get here. So this is again a big process. It depends upon the samples. It will take 1 hour to 2 hours. Okay. So after doing all the procedures, if you want to preserve the sample, you can keep the sample in minus 80 degrees centigrade refrigerator. This is a very big one, minus 80 degrees centigrade. Generally we use the refrigerators, they are up to minus 4 degrees. Okay, but this one is completely different, only for the samples, minus 80 degrees. Then this one is called biological safety cabinet, and they have to perform some tests here. You see the pipettes, rags, everything, reagents. The technician has to sit here, and they will open the door of this safety cabinet. Only they have to keep hands inside, nothing else. Okay, and then they have to do the test. That is the biological safety cabinet. Next we'll move to another room in RT-PCR, and one more room here. Here you see minus 20 degrees centigrade refrigerator. You can preserve kits for different tests here. And here you see another machine is laminar airflow. Laminar airflow cabinet here also, which is similar to the biological safety cabinet, but somewhat different. This is the PCR machine. We have to, after doing the procedures of all the samples what I have shown you, after that we have to keep inside. Okay, keep inside, and then it starts again. It will take 1 hour to 2 hours depending upon the samples, and after that you read the results in the computer. So that's the procedure of the COVID-19 RT-PCR test. So actually very big procedure, and there we'll get positives and negatives. Here one more system. Okay, this is the doffing room. After all the procedures, the technician can remove the personal protective equipment. Okay, I have shown the first one. First one is the donning room here. So donning room where they will wear personal protective equipment and do the all procedure, and finally after finishing the procedures they have to remove personal protective equipment in this room, doffing room. just hand wash area. These are all some sanitizers and antiseptic lotions. So here there are instructions how to remove dwarfing PPE. Okay. This one is biomedical waste room, where you see some bins to dispose the waste, biohazard waste. And this is autoclave. After removing the PPE and everything, they have to autoclave it. Autoclave is nothing but a sterilization procedure. We just remove and just dispose the waste material is not good, which is hazardous, not good for health for anyone. Okay, that is called biohazardous material. Before disposing, we have autoclave it for the sterilization. If any other microorganisms or viruses are there, they will die whenever we do this procedure. So finally we just dispose it. I hope you understand this. And this is my room. So my room annamata. And these are the other faculty rooms. I don't want to show them because I don't want to disturb their privacy. That is other staff rooms. This is just a small office room. Okay, it's a bit of medical college campus that you are watching. I think you like this. It's so cloudy, and yesterday there was a heavy rain. Okay, so actually these are the boys hostel and girls hostel. So what you're expecting from this channel and what you want to see more in this channel, please comment below. And if you like my videos, don't forget to like, share and subscribe my channel. See you soon. Take care. Bye.